Hey guys! That doesn't sound natural. Hey friends! Welcome to our third video for Unit 3, Sensation and Perception. We've watched 3.1, you watched 3.2, and now I'm back to give you 3.3 and 3.4, which is vision. So we're talking all things about the eye, the anatomy of the eye, how we're able to see, different colors, why some people can see all the colors and some people can't. Let's get right into it. Okay, here are the learning targets that we're going to look at, topic 3.3 and 3.4. So let's go ahead and get started. Okay, here is the eyeball. All right, we've got all the parts that we're going to look at. So we have the cornea, that's where we're going to start. So this is the transparent outer covering of the eye. So that would be this shape right here. Then we have the iris. This is the muscle that controls how much light enters the eye. So it's going to either expand or contrast. And that leads us to the pupil, which is an actual hole in the eye that allows light to enter in. So depending on the size of your pupil, tells us if the iris is letting a lot of light in or not a lot of light in. So the pupil is actually a hole. I need you to know that. It's not a black dot in your eye. It's an actual hole in, and that's how your light is able to enter the eye. Your iris, like I said, is the muscle, but it's also the colored part of your eye. So if you have blue eyes, green eyes, brown eyes, that's the iris that you're seeing. Then behind that, we have our lens, and this focuses the light onto the retina. And so if you have an issue with your lenses, this is probably like why you have to wear glasses or contacts. But as it focuses the light on the retina, the process of that is called accommodation. So let's talk about the retina. That's this whole back area. This contains all of our visual sensory receptors. We're going to dive into that, but we saw in the retina, that's where transduction happens in vision. And the retina is made up of a lot of different receptors, and we're going to dive into that. The fovea is going to be right here in the middle of the retina. This is the point of central focus. So this contains most of the eye's cones, which we'll talk about, but this is where your vision is the clearest, the most precise, and it's right in front of you. So wherever you're looking, right in front of you, that's your clearest vision because that's hitting your fovea. And then all of those neural impulses that get sent from the retina, they get sent to the optic nerve, and that takes the message to the brain. And then you have a blind spot. So where all of the receptors kind of come together to create that optic nerve, you have a blind spot. There's no receptor there. And we're going to do a blind spot activity to show you that you actually do have a blind spot. You just don't notice it because like we said, your mind does not like gaps, right? So your mind likes to fill in the blanks, and you have two eyes, and one kind of helps the other one out. But I'll show you. You have one, even if you don't believe me. Okay, let's talk about the retina because this is a really, really important part of the eye. So we have the light-sensitive inner surface of the eye. So it's inside, right? Like I said before, it's in this back area of the eye on the inside, containing receptors, including rods and cones, which we'll talk about the difference of those, and other layers of other neurons containing our bipolar and our ganglion cells that help us process our visual information. Light travels to the back of the retina first. So back, it's going to hit those rods and cones first, which are your photoreceptors. It's going to go all the way to those first, and then it's going to move forward to then hit the bipolar cells and the ganglion cells, and then those ganglion cells move that message to the optic nerve, and that takes it off to the brain. So hits the back of the retina first and then works its way forward. The fovea, like we said, is a central point of the retina within which the eye's cones cluster together. So if you're looking at the back of the retina, which we're going to draw this out too to maybe help you a little bit, but our fovea, we said, is our clearest part of vision, and that's where all of our cones are. So all of our cones are clustered together in our fovea, and that allows us to see our clear vision and our color vision. Cones allow us to see color. I like to think of C in cones for see in color. Cones tell us color. All of our rods are on the outside. These are the photoreceptors that allow us to have peripheral vision. So you know how you're able to look straight ahead and you're seeing something clearly, but you're also seeing stuff on the sides? You can thank your rods for that. And they allow us to see in black and white. So you can see in the picture that fovea is right in line with the pupil, the lens, because that gives us our clearest vision and our color vision. Okay, let's talk about the difference between nearsighted and farsighted. So some of you might be familiar with these terms if you have glasses or you've gone to a vision doctor, but nearsightedness is a condition in which nearby objects are seen more clearly than distance objects because the eye is elongated in shape. So the image focuses before it hits the retina. So you can see in our little diagram here, you've got your light and your visual information that's coming through the eye, and it's going to focus in front of the retina. So this allows you to see things up close more clearly, and it has to do with the shape of your eye. So you can see in our circles up here at the top, the shape of a typical eye is very round. Shape of a nearsighted eye is going to be more of an oval shape or elongated, so more this way as opposed to like this. Okay, so normal vision, someone who has 20/20, it would look like this picture is very clear. You see both of the children and both of the balls that they're holding. Or if you were to have nearsightedness and you were to look at the picture, you'd see that the boys in the back are a little out of focus, but you're able to fully focus on this ball in the front because it's close to you. Okay, farsightedness, so the opposite. It's probably going to be different, right? So a condition in which far away objects are seen more clearly than near objects because the eye is shortened and the image focuses after it hits the retina. So we have, again, our shape of our typical eye, and we have our shape of our farsighted eye, which is shortened. So nearsighted was elongated, farsighted is shortened. And you can see that light in all of the visual information is coming in, and it's focusing behind the retina as opposed to in front. Okay, so let's look at this picture here. We can see that our far-sighted objects, so these flowers in our trees, are a lot more visible than that book up close. You can see that that book up close is very blurry, as opposed to with normal vision, you'd be able to see the trees and the flowers in the back and be able to read the pages. So a lot of times, this kicks in when we're older. I don't know if like your parents or your grandparents, if you've ever seen them like put their reading glasses on, that's because when they put something up close, it's hard for them to see. You might see them like put it all the way out here, so maybe they can read it, or they put their reading glasses on. Okay, let's look at this. So this is a more detailed picture of the retina and the different cells that are in there that help us process our visual information. So like we were talking about, we have our rods and our cones in that first section right there. Our cones are going to be different than our rods, obviously, and we just talked about in the fovea that our cones are going to be more clustered together as opposed to our rods are going to be more spread out and more on those outer edges of the retina. This is going to tell us colors, black and white, our clear vision versus our peripheral vision. And we can see, remember that the neural impulse or that visual information comes all the way back to this first column first and then works its way to the second and third column. So that second column is the bipolar cells. I like to think of bi as two, so they're the second group. And then our ganglion cells. I had a kid my first year teaching called them the ganglion cells, and he always remembered it that way. I don't know how that helped him, but I can't ever teach this without thinking of ganglion cells, but it's ganglion. And these send that information to our optic nerve, which then takes it to the visual cortex. So we can actually interpret what we are seeing. Photoreceptors, so specific are rods and cones. Rods are sensitive to light. Cones are sensitive to color and fine detail. In this image, notice what the next cell that the rod connects to, vice versa the cones. What is different? Okay, bipolar and ganglion cells. Bipolar cells receive messages from photoreceptors and transmit them to the ganglion cells, which then form the optic nerve. Cones each have their own bipolar cell, and multiple rods will share one bipolar cell. So their message is not sent as clearly. So make sure we notice that. If we can see our rods, we've got like 3 that filter into one bipolar cell as opposed to one cone goes to one bipolar cell. So that allows for our vision, that allows for the cones to give us that clear vision that we were talking about. Okay, let's look at the different areas of visible light here. So we've got our infrared that we're not able to see, or our ultraviolet rays that we're not able to see, but we're able to see all of these colored light waves here in the middle. So red, orange, yellow, green, blue, indigo, and violet. Almost forgot what I stood for. So we have our hue. So this is the dimension of color determined by wavelength of light, right? So we can see that our red is going to have a longer wavelength as opposed to our violet, which is going to have a shorter wavelength, and that's going to help us determine the color. So I don't know if you're ever like on Lightroom editing your pictures and you go to edit the specific color and you click on where it says hue. That's where that comes from. Intensity, the amount of energy in a wave determines its amplitude. So how high each wave is related to the perceived brightness. So if we have a higher amplitude, it's going to be a brighter color, as opposed to a lower one, it's going to be a duller color. Just like you can see this, the background on this PowerPoint is kind of a light, dull blue, as opposed to the blue that's in our visual light spectrum over here. So those would have different intensities. Test your blind spot. We're going to do this in class. Okay, pathway from the eyes to the visual cortex. So let's do a little review here. Remember that information has to get sent through the thalamus and then it goes to the visual cortex. So you can see we have our eyeballs up here in the front. We have our retina, which is the back part of the eyeball. And that visual information has been passed through the rods and cones, bipolar cells, ganglion cells, and those ganglion cells kind of curl together to make that optic nerve. That optic nerve sent to our thalamus first. Remember the like the airport of your brain or the operator of your brain. It's got to go there first before it determines like where it's supposed to go. And then that information gets sent back to our visual cortex. And remember, some parts of vision go to one hemisphere versus the other, and it just depends on what we're looking at, right? Are we looking at more spatial shapes? Are we looking at face recognition? What are we looking at? And so that information from each of our eyes is going to get sent to the correct hemisphere and the correct part of the visual cortex in the back in our occipital lobe. Feature detectors. What does this sound like? Your ability to detect features, and that's exactly what it is. So we have nerve cells in our visual cortex. So we just saw that on the brain, it's in the back, that respond to specific features like edges, angles, length, and movement. You're able to detect those things when you're looking at something. There are even some feature detectors that are specifically sensitive to the human face. So you're able to detect certain features on the face. Surprise, your brain does a lot of things. Would feature detectors be sensory or perceptual experience? Would it be a sensory or perceptual experience? What do we think? So we have our visual information processing. Processing several aspects of the stimulus at the same time is called parallel processing, because you're You're processing all these different things at the same time. So parallel processing. The brain divides a visual scene into subdivisions like color, depth, form, and movement. So you're seeing something, but then the brain is dividing it up into all these different categories at the same time. And you don't even realize that you're doing it. Your brain is doing it for you. So you see something, we have our retinal processing, right? So that idea of taking that information from the retina to the brain. So our transduction is happening here. We have our feature detection. So you're able to detect certain features, whether that's movement, angles, shapes, or facial features. All of that is happening at the same time. You're detecting different features while also detecting color and a bunch of different things. And then put all of that together, you're able to recognize or perceive what you're actually looking at. Okay, now, theories of color vision. How are we able to see in color? Why can some people only see some colors? Why can some people only see in black and white? What does that mean? Let's talk about it. Okay, so we've got one theory first. We've got the trichromatic theory. You might see it as the Young-Helmholtz theory. Those are the two guys that put it together. So based on behavioral experiments, Helmholtz suggested that the retina contains 3 receptors, so 3 different cones, sensitive to red, blue, and to green. And any other color that you see after that is a mixture of those put together. Now, sometimes we have some color deficiencies or color blindness. Some people will say, I'm colorblind. If you're able to see color, you're not colorblind. You just have color deficiencies, meaning you can only see some colors and not all of the colors. So this is a genetic disorder which prevents individuals from discriminating between certain colors due to a weakness in or lack of one of the cones. So we have our little circle off to the side. It's got some different colors and it has a number on the inside. Try to see if you can pick out which number we're looking at. Also, same thing with our apples down here. At the top, someone who does not have a color deficiency can see a red apple and a green apple. But someone who does have a color deficiency, these apples might look similar. One might look slightly darker, but you can see that they're both a yellow tint. The most common form of color blindness or a color deficiency, right, is the difficulty distinguishing between red and green. So this is why people need to make sure that if you have this color deficiency, that you understand like the placement of the lights, like on a stoplight. You know, is it red first or green first? We know yellow's in the middle, but what is it, red first on the left? Is it green on the left? We want to make sure that we know the order, because when those colors look the same, we don't know if we go or stop, right? And that could be a life or death situation. Complete color deficiency does exist, but it's rare. So this would be like everything's in black and white. Couldn't see any color. So it'd be like watching a black and white movie, but that would be your life. Okay, we've got another theory. Remember, in psychology, you got all the theories. Okay, there's never just one. This is the opponent process theory. So we've got Herring. He proposed that we process 4 primary colors opposed in pairs of red, green, blue and yellow, black and white. So basically, you have photoreceptors that pick up on these colors, and once you like wear one out, you're able to then see the other one. And you're probably like, what does that mean? I'll show you. We'll do an example here. So after imaging, so this is going to go along with those theories. So remember which pairs go together. Blue and yellow go together. They're in opposition with each other, but they're like grouped together. Green and red, white and black. Now look at this picture. We're going to look at it for 30 seconds, or pause your video here. I want you to stare right in the middle. Don't move away. Look at it for a minimum of 30 seconds. Now, as you switch to the white screen, unpause and switch to the white screen, what do we see? Hopefully, you now saw what the American flag looks like with the correct colors. Okay, so where there was yellow, hopefully after you stared at it for a while. So while you were staring at it, those photoreceptors were firing yellow, black, and green. Yellow, black, and green. Like over and over and over again. Those are the only colors that we were focusing on, right? And as soon as I switched to white, those had been worn out, so you saw their opponent. So that's where you saw the blue, the white, and the red. Does that make sense? Do we see how they're paired together like that? Crazy. Okay, so we're going to talk more in depth when I see you next, but make sure you have filled out your notes for this video. We're going to draw the eyeball together. We're going to label all of the things to get some extra practice, but you really want to make sure that we understand the different color theories. We understand what a color deficiency is. We understand all the different parts of the eyeball and what they do, why are they there. And remember where transduction happens, which is in the retina with those photoreceptors of rods and cones. Okay, I'll see you next time, friends.